Hello everybody, today we are going to do the wet lab portion of the Carolina kit, DNA chips, genes to disease. It's not one of the SCMEs that are part of the initial large project. However, there is still an application to where you can use this and jump into the actual SCME kits. Okay, so right here we have the main stars of this little production. The genes, gene 1, 2, 3, 4, 5, and 6. So what we are going to do is we are just going to carefully add a drop of each of these onto our slides. Now the slide is representative of a microarray. Normally microarrays can come in like 96 well plates or larger and typically have different sets of genes. So we're going to apply the first gene. Just a little drop will do. Got a lower drop. One of the things about this kit though is all the genes you've seen up here, they all come in a wax form, so they do have to be submerged basically in boiling water. If not submerged, then at least dipped inside of them just to get the wax into liquid form to where you see. Okay, now that we have all 6 of our genes onto the plate, the next thing we're going to do is we're going to add a little bit of the hybridization buffer. Now this buffer is pretty caustic and may cause burns, as you can see here. So you do want to wear gloves. You always want to wear gloves while you're in the lab and working with anything. So what should occur is we should see a color change a little time after adding the little drops. You can even see some of the color starting to form right there for gene 1 and 2. Okay, so now that we've waited long enough for our little spots to dry, you can see that it does look a little different from when it was originally dropped in, the hybridization buffer. Number 2 doesn't look as purple anymore. Instead, 2, 3, and 6 look like they're all the same color blue. Maybe the brightness changes a little bit. Spot 1 is a little bit hard to see, but it is a color pink. Spots 4 and 5, they are clear. 5 looks like it may have a hint of color, possibly a little bit of purple. You never know. In an actual DNA microarray though, you don't actually ever see the color because the cDNA, complementary DNA that it's supposed to represent, is actually so dilute that once everything is said and done, everything's mixed, there really isn't a way to see the color with our eyes. That's when we use the actual more sophisticated devices that can measure everything for us. Okay, so right now we're gonna be working on the paper portion of this lab. The paper microarray analysis actually. There is a wet lab component, we can get to that. But just to visualize it without actually doing anything in the lab, we have these sequences. These are going to be the complementary DNAs from normal cells. Now what you see, you have your 5' to 3' or 3' to 5' ends. And on this other side, you have the complementary DNAs from abnormal tumor cells. So what's going to happen is, the main idea is that all these would get cut up into individual pieces. Maybe even copy this on blue paper, copy this side on red paper. And then right here, our paper microarray is going to contain a sequence of DNA right there, that whatever is on here, whatever little sequence, maybe it's this one, maybe it's this one, maybe it's this little piece right here. that is gonna match completely up to this, maybe this. It could bind onto both of them. And what that means is the complementary DNAs will not bind to any of these if they are not an exact match. When they are an exact match, we can further determine the mRNA that the cell is producing. For instance, if it was, say, red, if this spot was all red, then that would mean that the cancer cell is actually expressing this mRNA over what a normal cell would be expressing. If this one were, say, purple, which is a mix of red and blue, then that means the cancer cell and the normal cell would be expressing the gene at the same time. So between purple and between a red cell, you could say, it could be determined that this gene is more active in cancer cells as compared to this one, which is active in both of them. However, this one could potentially be cancerous. This one, for example, may actually be blue, and that would mean that the cancer cell is not exhibiting it. So the main idea behind the DNA microarray is that we have all these genes both in cancer cells and in normal cells, and really what we're trying to determine is what's going on. Like, is the cancer cell producing more mRNA that produces a certain protein more than the original cell, the normal cell, or is there something about a certain cancer gene? Could be this one, gene 6, that enables cancer to be so effective, so able to grow and evade our own immune system. Maybe even gene 4 is the one that keeps it alive and prevents it from essentially killing itself, apoptosis. In an actual real microarray, we wouldn't just have 6, of course. We would have thousands and thousands that would bind, and we wouldn't actually be able to see the colors to any of this. So in conclusion, we have the paper microarray set, and then we also have the wet lab portion of it. These are all just representations of an actual microarray. These microarrays contain thousands and thousands of wells, and more often than not, we actually don't see the color that's in them. Each one of these genes, for instance, we have 6 right here compared to the thousands. We can take this gene, corresponds to that gene. That one was pink. This one would end up being pink and bind to one of these, right? Now, the cDNAs shown previously right here, these would all have to have some part of it that matches exactly to one of these. And when that happens, in this lab, we get a color. We get red for when a cell is expressing mRNA that creates a protein and is cancerous. And if it were to be blue, though, that would mean it's a normal functioning cell and it's actually needed for its survival or daily activities. The main purpose of a DNA microarray is we have all these genes and we know essentially what they do, but what we don't know exactly could be that what's being expressed in a cancer cell versus a normal cell. If gene 3 is expressing something in a cancer cell, say a mechanism which the cell uses to escape cell death, to evade the immune system, it's very possible that it could be used to target for future medicine, develop some kind of cure, or essentially some kind of drug that can block gene 3. Gene 4, for example, that one could have been blue, in which case it would just be a normal protein that's being expressed. If it were purple, for example, that would mean both the cancer and the normal cells would be expressing a gene that created a protein that they both needed. So in other words, it's something that maybe wouldn't be as wise to target for drug creation.